My name is Robert Noveski. I'm the founder and CEO of Sinceritas. We are a pharmaceutical company focusing on cannabis-based medicine. So today I will talk about the challenges of cultivating and growing pharmaceutical-grade cannabis. It's a big topic. I could talk hours about this, but now I have to shorten it in 30 minutes. Let's see what I can bring over in 30 minutes to you guys. So basically, producing cannabis flowers according to pharmaceutical standards basically requires several significant challenges, or possesses several significant challenges. These challenges arise from the need to ensure consistent quality of the product, safety, and efficacy of the product. That's like every other pharmaceutical product. Additionally, as a producer, we have to follow certain regulation and be compliant to certain guidelines. Here to be mentioned actually the guideline on good agricultural and collective practice for starting materials of herbal origin. Also the EU GMP Part 2 basic requirements for active substances using as starting materials. The EU GMP Part 1 Annex 7 manufacturing of herbal medical products. Here to mention actually depends, especially in Germany, every state sees the product differently. So you might fall under the EU GMP Part 1 or Part 2, depending how the state in Germany sees it. So it can see it as a API, then it's Part 2, and if it sees it as a herbal medical product, it's Part 1. Additionally to this, there is the ICH topic Q7 good manufacturing practice for active pharmaceutical ingredients to be followed. Also the guidelines on principles of good distribution practice of the API, or the guidelines of good distribution practice of the medical products for human use. If the product doesn't have a monograph, so basically there is the Committee of Herbal Medical Products to be followed. So, come on, not working. Something is stuck. Okay, fine. So here can we see a table actually of the activities done in a facility and which guideline has to be followed. So we can see, for example, the cultivation, collection, and harvesting of plants, algae, fungi, lichens, and collection of exudates has to be under GACP. Basically, the cutting and drying of this has to be under GACP. But if the product is basically the dried product as a finished product, then the drying process has to be under GMP Part 1 or Part 2, like said before. All the other processes like the distillation, extraction, and so on has to be under GMP Part 1 or 2. And further processing into the dosage forms or also the packaging as a medical products has to be under GMP Part 1. So if you apply this now to the pharmaceutical cannabis, we can say that the cultivation of the mother plants, the propagation, vegetation, flowering, and harvest falls under the GACP guideline, and the drying, trimming, and primary packaging falls actually under GACP guideline. The product has to meet some certain specification to ensure a certain quality. Here for the cannabis flowers, we can say actually in this table you can see the parameters. There is a specification according to the Deutsche Arzneimittelbuch 2018 and the new specification according to European Pharmacopoeia 11th edition, which is effective I think from this month on or from next month on? Next month. It's next month, I think, the 1st of July, okay, from next month on. So you can see the parameters, basically properties, identification, so you have to see if it complies, actually, if the flower is a flower, not something else. You can see the foreign matter has to be less than 2% in both specifications. The impurity, basically the CBN, has to be less than 1%. The loss on drying, so basically the moisture content, according to the DAB specification, has to be less than 10%. and according to the new specification, there's a change. It can be up to less than 12%. So there is a bit more moisture, room to be the flowers to be more moisture. In terms of the assay, so basically cannabinoid content has to be 90 to 100% of the declared concentration in both cases. In terms of the aflatoxins, you can see the B1 has to be less than 2 microgram per kilogram in both cases. For the aflatoxins in B1, B2, G1, and G2, it's less than 4. Heavy metals, we can say actually that cadmium, lead, and quicksilver, mercury, quicksilver, has to be, you see the concentration, the 1, 5, and 0.1 ppm. To the new specification, there is the addition of arsen, and the concentration of what was this now is reduced to 1. The concentration of, which one was it now? I need my glasses. So basically, PB, basically the lead is down to 1 ppm from 5. Then there is the residual, basically pesticides. So of course there has to be non-pesticide. So there's very strict limits in this. In both cases, the microbiological purity, total aerobic microbiological count has to be less than 500,000 colony forming units per gram. The total yeast and mold count has to be less than 50,000 colony forming units per gram. In both specifications, the biotolerant gram-negative bacteria has to be less than 10,000 colony forming units per gram. Of course, there has to be an absence of Escherichia coli bacteria and Salmonella. So, coming now to the cultivation part, we can say basically there are certain processes and things to be followed to be able to cultivate pharmaceutical cannabis grade A. So here is an overview summary. I will talk later a little bit more in detail about every of this topic. So the first one is basically the genetic selection. So you will need to have a strain development process with the phenotype selection. You will need to have a life cycle management of the mother plants. If you use mother plants for propagation, if you use tissue culture, it's a different story. You need to have a growing environment where you have constant and uniform temperature, humidity, CO2 supplementation. The grow media and nutrients, you need to have a good drainage and aeration. And you have to adjust the nutrient mix according to the growth stages. So in terms of watering practices, very important is the quality of the water and the dryback period of the grow medium. Depends what kind of grow medium you use. Cultivation techniques, to have basically, from our experience, we use the screen of green because this gives us a uniform canopy. Every plant is the same height, gets the same intensity of light. So you have a uniform light exposure also. Very important is the pest and disease management, which usually people don't put so much efforts, but it's a very, very important point. So it's very important to have a strong cleaning procedures, that the environment is always clean, disinfected, and a regular plant inspection. So it's very important to check the plants if there is something on the plant visually, but also under the microscope, do some testings. Flowering and harvesting, it's very important to basically meet the right harvesting time. This is very strain specific. What you can say is basically sativa strains need a bit more longer flowering time, where indica and hybrid need less longer flowering time. And last but not least, the harvesting process. Post-harvesting process is basically very important to have an optimal drying and curing process with good conditions to maximize the flavor and the aroma of the flowers. Coming to the single points now. So basically the new strain development phenotype selection, what is it actually? So it's very important to choose from a certain basically strain the right phenotype. Why? We want to know for this basically certain strain to have the peak cannabis concentration during this strain development process. We determine at the peak cannabis concentration. We also check for the duration of the peak concentration. So how long this THC, the longer I keep it in the flowering, how long the THC level is kept before it drops. So there is an effect of basically where the plant produces biomass and THC. Depends which process dominates, will basically determine the THC content in percent, because the percentage is relative, not an absolute level. So very important is also the morphology of the flowers. So different phenotypes of a certain strain can have more dense flowers, more fluffy flowers, bigger flowers, smaller flowers. It's very important to choose the right flower. Why? Because the microbiological purity also depends on the morphology of the flower. You can see usually that bigger flowers and more dense flowers have a bigger risk of microbiological contamination than smaller flowers and fluffy flowers. On the other hand, the clients want big, dense flowers. So there is a bit of a kind of a very important thing in the strain development process to find the optimum of both. The terpene profile, so that means basically the terpenes and the THC is not produced in parallel, so there are different processes with different timing. So you have to see actually what is the optimal basically phenotype to have an optimal THC level, but also an optimal concentration of terpenes. And of course, with the strain development process, you determine at the harvesting time. So on the right side of the table, you can see one strain in different phenotypes. On the x-axis, you have the days in flowering, and on the y-axis, you have the THC concentration. So you say after, so we started the testing at the 34th day of flowering, and you can see for the different phenotypes the THC concentration. So there is one phenotype which has very low concentration from the same strain, but other phenotypes have higher concentration. And now you can see over the time how the THC level evolves. What you want is basically something where you have an upward sloping curve with very constant the longer you keep it, because the terpenes are built on the longer you keep it. So it means the red curve is a very optimal phenotype, where you can say actually the lower one, it's not so interesting because the THC level is very low, and there are some phenotypes where the THC level is dropping, not so much, but still dropping. So these phenotypes are maybe also not very optimal. Of course, you have to check again for the morphology of the flower and so on. So you have to follow, you have to have a process to optimize all these criteria. So what else to say about genetic selection? What you can say is actually genetic uniformity is also batch homogeneity. Choosing the right genetics and also the right phenotype gives you homogeneity within the batch. So what you can see actually here, what we did is within one batch, analyze 24 plants and check the THC content. You can see very well if you select the strain and the phenotype very well, you can see that pretty much all the plants are within the range of plus minus 10%, are quite uniform. So you don't have much dispersion in the THC content. This is a very, very important point, especially if you do later on a stability study on your product. If the points vary too much, you might have a problem where in the first T1 you take a high THC, and a T0 take a high THC, and T1 you get a low THC and you're out of spec in your stability study, and you can drop your stability study, or the product will have a very short shelf life. So it's very important to have a very uniform batch. This guarantees you to have a stability study which gives you also long shelf lives. Our product, how we package it, have a shelf life up to 2 years, and they are stable for the whole 2 years. Not only because of the genetic selection, but also the way how we package it and so on. Also very important is basically to define the batch size based on the drying room, but also on the flowering room. So what you want is basically not to overpack your flowering room, because this gives you basically problematics in regards of powdery mildew and other microbiological contamination, but also you don't want to have too little plants, because you want to optimize the yield, the electricity and the labor you spend. You want to get something out of the room. So here what you can see, also very important stuff in the genetic selection, or basically the selection of the genetics, is basically the life cycle management of the mother plants. What we can see, and we tested this, every strain or phenotype has a different kind of behavior in terms of how the THC content is from the clones taking from the mother plant according to the age. You can see the mother plant after 6 months, the THC, if you take clones, the THC of these clones is dropping. So it means basically the genetic is not so stable. So you need to renew the mother plants after a while. So usually after 6 months, you have to renew the mother plants. Some, it's definitely strain specific. Some mother plants have a longer period, some shorter. If you choose a very bad one and you didn't test it, it can drop even very quick. And you will lose your genetics. So you can start from the beginning. Growing environment. So what can we say there? So of course you need to have a controlled environment, preferable it's indoor. Some people do greenhouse or outdoor. Outdoor is very difficult to control the environment. Greenhouse is possible, but also difficult. Of course, the indoor is the most easiest one to control the environment. So you have to optimize the temperature, humidity, CO2 supplementation, and the light intensity to have an optimal growth. We actually grow with the temperature of the rooms between 23 and 26 during the day phase, and cool it a little bit down during the night. The humidity, we keep it between 40 and 60 during the veggie phase, and 30 to 40 during the flowering phase. Especially in the last 2 weeks on the flowering phase, it's important to keep the humidity low to make sure that there is no microbiological contamination or mold grows always with higher humidity. So also very important to maintain the parameters constant. The plants doesn't like if the humidity is fluctuating too much. Powdery mildew is especially the problem then. Doesn't like also if the light intensity fluctuates too much. In general, the more homogeneous you keep the parameters, the more homogeneous batch you get from batch by batch basis. So we have a strain which we cultivated now maybe 30 times, and you can see that the THC level from the batch to batch is always homogeneous, but also within the batch is homogeneous. So it gives you a good genetic. What we do is also we monitor the vapor pressure deficit, so how the plant is basically evaporating the water. What you can say is in an early stage of vegetation, you want a lower vapor pressure deficit, so it means the plant is keeping the water. In a later stage, you want, in a flowering stage, you want higher VPD levels, that the water is evaporated from the plant and not kept into the plant. This is also a point where you can have microbiological contamination because if the VPD is too low in a late stage. What can we say about the grow media and nutrients? So a good drainage and aeration is very important for the roots also. So you can say actually that the nutrient mix recipes is adjusted to the growth stages. You can say higher nitrogen concentration during the vegetation phase where the plant is growing, needs nitrogen. Later on in a basically flowering phase, you need more phosphorus and potassium to develop the flower. What you can say is the more phosphorus and potassium you put, the more dense the flower will become. So if you pump them with a lot of phosphorus and potassium, you get very dense flowers. Again, you have to make sure that you don't make them too dense because this can have an effect on the microbiological contamination. So this is a way how you have to find an optimum there. Very important is also to have a calibration curve of the nutrients based on the concentration in its sea. So, and to calculate exactly the nitrogen, phosphorus, potassium concentration, what you input into the plants per watering. So what you do is you take the spec sheets of the nutrients, see what's inside, do the calculation, what you put into the plant, and measure this in ppm, that you know what is the nutrients you put into the plant. You have to maintain also a good pH level during the grow media between 6 and 7 for the optimal nutrient uptake. What you can say is actually if the pH level is higher or lower, certain nutrients cannot be uptaken anymore. So, and the plant will get some deficits. You can see it in the leaves that there is some deficits, but usually you put enough nutrients inside, but the plant is not able to take it up. So therefore, it's very important to check the pH level. We monitor the pH level and EC level in the grow media as an indicator for the condition of the plant. So what you want to know is basically how much nutrients does the plant take and how much is still in the medium. This gives you a good indication if the plant is healthy. Watering practices. So what we can say is there is basically very important thing is the quality of starting water. So preferably you use reverse osmosis water. This guarantees you that it's free from heavy metals, pesticides, or microbial contaminations. So you don't want to water your plant with bad water because this will affect immediately the quality of the plant. The watering cycles are also very important depending on what kind of grow medium you use. You have more or less watering cycles. We use rockwool cubes where we have up to 5 watering cycles per day. If you use basically other grow media like coco perlite or super composted soil, you have less watering cycles because the media has a capacity to keep water and nutrients. It's a buffer capacity where the rockwool cube doesn't have so much. So we measure also the volume of nutrients per solution per plant. So like again, so it's always to see actually what you put and what the plant gets to monitor this and allow also the topsoil to dry out between the waterings. It's very important, especially if you have a rockwool cubes, that there is a dry back period. Cultivation techniques. Many techniques actually how you can cultivate the plants. So some people let them grow like they are. Some people do sea of green. We use actually a scree of green. Actually, why? Because this gives us a uniform canopy. Basically, all the plants are the same height and all the buds are on the same height and get the same intensity of light. So through regular pruning, basically by removing the leaves, small branches, we direct the energy to the top flowers and all the smaller one we take out that we have uniform flowers. This gives us also a batch uniformity. It's very important also, like I said, especially if you do later on stability studies, that you have a longer stability shelf life. Wait a second. So I wanted to show this. Okay. So what we tested also is basically the effect of light intensity on the THC concentration. In general, what you can say, less light, less THC. That's a general, basically, outcome of this. But also, if you give too much light, you can have some side effects. Light, if you give more light, you have to give also more CO2, basically, that the plant can uptake this light. Pest and disease management. So it's very important to have an integrated pest management system. So basically, you have a known pesticides policy, of course, because this will affect immediately the specification. It will be tested, and you have it in the test results. Very important to have a strong cleaning procedures and rigorous hygiene, especially also by for the equipment and the people. The biggest risk for the plants are actually the people who enter the facility. So it depends actually where the facility is. You have different risk. Is it more the facility, let's say, in an urban area in a city? You have certain risk, but if it's outside somewhere where there's a field around and you have field workers who come and work also in the facility, you have different risk. So it means basically you have to exactly check where is your facility and what is the surrounding of the facility to determine at the risk actually what can happen to you. Very important is also the regular plant inspection to check the plants if the plant has some deficiencies, if the plant is basically attacked by insects or some other viruses. Also a very big topic lately. The hop lantern virus actually is spreading around, so it's also very important not to introduce clones or genetics from suppliers you don't know, you don't trust, you don't qualify. This is the biggest risk to your facility. You can bring some clones, and the clones can be contaminated by all kind of insects, root aphids, spider mites, broad mites, trips, and so on. Flowering and harvesting. So like I said, it's very important to determine at the right harvesting time. This you do through the strain development and the phenotype selection, what I said in the beginning. This gives you actually the consistency of the cannabinoid for every batch and the uniformity. So this is of course strain specific. What we also have is in-process control during this period of growing. So we can evaluate basically through a microscopy of the trichomes when is the right time to harvest. In general, what we have is basically once we develop the strain, stabilize the strain, usually the strain has always the same day of harvest. It can vary 1 or 2 days, but it's very, very stable. So if you have 45 days, it's always 45 days. If it's 48 days, it's always 48 days. It can vary 1 day, but it's not like it varies 5-6 days. So once you stabilize the strain, you can put it in a plan and you can be pretty much sure it's the right moment to harvest. And you can check it by the in-process control, but it gives you it's always the right timing. What we also test is basically cannabinoid content over the growing period and compare it to our strain development process and the curve, if it follows the curve what we had in the development of the strain. So you can see if something is going wrong and you can act very early. Same for the microbiological contamination. We test it many times, not only on the end. So during the life cycle of the plant, during the flowering, we test many times the microbiological contamination and check if it's still in line or if something is growing to act very early. Post-harvesting process. One of the most important processes actually in producing high quality cannabis is basically the drying process. Here it's very important to have a validated drying process and a good drying room. So it's important to reduce the moisture of the plant as quick as possible, but not too quick to make it too crispy outside, but still moist inside. So what we have is basically a validated process. We have a 2-step drying process. We hang dry the flowers for a certain amount of time, and then we cut them from the stem and put them on trays and finish the drying in trays. Like I said, it's a very critical step. So it's very important to detect wet spots in this process. So what we did is, for example, here you can see on the chart, once you put it on the trolleys, you can see the number of the trolleys, the position of the trolley, top, middle, and bottom, and the moisture content. You can see that this is quite homogeneous dried. So independent where the flower is, it's always pretty much the same moisture content. So that's very important to optimize basically the drying parameters that you can achieve this. We use drying parameters, for example, of 15 to 20 degrees and the humidity of 50 to 60%. The drying can be from 7 days to 14 days. It depends on the strain. It depends also on the morphology of the flower. Usually dense flowers, bigger flowers dry longer. Smaller flowers, fluffier flowers dry quicker. So again, there you have to optimize this with the strain selection process to have an optimal, also good strain selection to dry it also good. You don't want to have too dense and too big flowers. Of course, the client or the patients want dense and big flowers. So here also you have to maintain a constant and uniform drying conditions. So of course the drying room has to be able to keep the parameters and not fluctuate. Last but not least is the proper curing process. So the curing is basically enhancing the flavor and the potency of the flowers. Here you can say can go up to 14 days or even longer. You want to reduce the chlorophyll in the flowers. You want to basically enhance with reducing the chlorophyll in the flowers, the terpenes, the smell is better. And of course, if the patient is smoking it, it has a much better experience in this case. What we did also in the post-harvesting process is basically in our drying room to check the microbiological testing in terms of homogeneity of the drying. So you can see actually on the x-axis again the trolley, the position of the trolley, and the microbiological contamination. You can see that there are some flowers which are a bit high, but they're still below the threshold. But in general, you can see it's quite uniform. So there is no big spots where you have big spikes of maybe where there was a pocket of wet flowers and mold was created. So to summarize, basically producing cannabis to pharmaceutical standards involves overcoming complex regulatory, biological, and technical challenges. And to be successful in this field requires a deep understanding of both plant biology and pharmaceutical manufacturing processes, along with substantial investment in technology, facility, and personnel. Especially here to say the personnel. So it's very important to train the people. They have to follow the standard operating procedures, and constant training of the people enhances the quality of the product. Small deviation of the standard operating procedures can result in a less good quality of the product. So I hope you enjoyed the presentation, and thank you very much.